All right, let's dive into the fascinating world of protein behavior. Today, we're taking a deep dive into the journey of a protein called superoxide dismutase 1. You might know it as SOD1.
SOD1.
Yeah.
Such a crucial enzyme.
It is, right? And it plays a key role in protecting our bodies from those pesky, damaging free radicals.
Yeah, SOD1 is like a master defender. It catalyzes the dismutation of a superoxide radical, turns it into something much less harmful, hydrogen peroxide and molecular oxygen.
Pretty amazing how our bodies have these built-in defense mechanisms. But what makes this deep dive really interesting is SOD1's connection to a devastating neurodegenerative disease, amyotrophic lateral sclerosis, or ALS.
ALS. You probably know it as Lou Gehrig's disease.
Exactly. And the research we're exploring today digs deep into how SOD1 can actually transform from this helpful protein to a potentially harmful one.
It's all about the aggregation process, how these proteins clump together.
Right. And get this, the research we're looking at reveals that SOD1 undergoes a liquid-to-solid transition.
Almost like water freezing into ice.
Exactly. That's such a great analogy. And this shift, this change in state, seems to be a key piece of the puzzle in understanding SOD1's role in ALS.
It's a pretty big deal.
So to kick things off, let's talk about how the researchers actually induced SOD1 misfolding in the lab.
In vitro, as we scientists say.
Right, in vitro. They used hydrogen peroxide to mimic the oxidative stress that cells experience in conditions like ALS.
Sort of like recreating that stressful environment in a test tube.
Precisely. And they specifically worked with AMPO-SOD1.
Which is SOD1 without its metal cofactors.
Makes sense. So how did they actually observe the formation of those SOD1 fibrils, those clumps of protein that are a hallmark of aggregation?
They used a clever technique called the theoflavin T fluorescence assay. You see, theoflavin T, we call it THT for short, is a dye that emits a strong fluorescent signal when it binds to these fibrillar structures. So when they added THT to their SOD1
The
sample,
more SOD1 fibrils were forming.
the brighter the fluorescence. And they saw a significant increase in THT fluorescence when hydrogen peroxide was present.
So they directly confirmed that oxidation was driving the formation of those SOD1 fibrils.
Oxidation was the culprit.
Fascinating. They essentially recreated the oxidative stress environment in a test tube and watch how SOD1 reacted. Now, before we jump into the oxidation part, let's talk about how SOD1 normally exists in cells.
Ah, yes. This is where the concept of liquid phase separation comes in, or LLPS for short. It's a bit of a mouthful, but it's a fascinating process.
LLPS. All right, break it down for us.
Okay, so imagine this. You have a crowded cellular environment, right? Lots of molecules jostling around.
Like a busy city street.
Exactly. And in this bustling environment, certain proteins, including our friend SOD1, can cluster together.
So they don't just float around solo.
Nope. They form these dynamic, liquid-like droplets. It's kind of like how oil forms droplets in water.
They form these little communities. I love that. So how did the researchers actually observe this happening with SOD1?
They used purified SOD1 tagged with a fluorescent protein called EGFP. Think of it like a little beacon that allows them to see the SOD1.
Makes sense.
They put this tagged SOD1 in a solution with polyethylene glycol, or PEG.
PEG. Why PEG?
PEG is really good at simulating the crowded conditions inside a cell.
Oh, I see. So they were able to recreate that crowded cellular environment in the lab?
Exactly. And what they saw was pretty amazing. The SOD1 readily formed droplets in this PEG solution.
Just like they predicted. So these droplets weren't just static blobs, right?
You're right, they weren't. The researchers observed that these droplets could fuse together, forming larger droplets.
Like merging bubbles.
And they could even recover their shape quickly after being disrupted. They were incredibly dynamic.
That's amazing. So they confirmed that SOD1 undergoes this process of LLPS, forming these dynamic liquid droplets in a crowded environment. But how does oxidation then push these droplets toward that more solid aggregated state?
That's the next part of the story, and it gets really interesting.
Well, I can't wait to hear it. But for now, let's take a quick break.
We'll be right back with more on the fascinating world of SOD1 and its journey from liquid to solid.
Stay tuned. All right, so we left off with this picture of SOD1 existing as these dynamic liquid droplets inside cells. But how does oxidation actually push it toward that more solid aggregated state that we know is linked to ALS?
Well, the researchers took those SOD1 droplets and exposed them to increasing concentrations of hydrogen peroxide.
So they're basically cranking up the oxidative stress.
Exactly. And as they increased the hydrogen peroxide concentration, they noticed something interesting. Those droplets, they started to grow larger. and they began to form clusters.
So oxidative stress, which, can come from all sorts of things like pollution or even just a poor diet,
Right, all sorts of environmental factors.
it can actually push these liquid droplets towards a more solid state.
Exactly. And this shift towards a less dynamic state, it was further confirmed using a technique called fluorescence recovery after photobleaching, or FRP.
FRP, that sounds familiar. Didn't we talk about that earlier?
We did. Remember, it measures how quickly fluorescence returns to a spot after it's been bleached away by a laser.
Right. And because those molecules in a solid aggregate are less mobile, the fluorescence recovery would be slower.
Exactly. That's precisely what they observed. Those clustered droplets, they showed much slower fluorescence recovery compared to the nice dynamic droplets that hadn't been exposed to all that hydrogen peroxide.
So things were definitely getting more solid-like.
Definitely. And over time, something even more interesting happened. The oxidized SOD1, it started to form these large, irregular aggregates within those clusters.
So unlike those unoxidized droplets, which stayed nice and stable and dynamic,
The oxidized ones were going through some major changes.
and they used THT staining to confirm the presence of these aggregates, right?
You got it. THT, remember, it loves to bind to those aggregate structures.
And it emits a fluorescent signal so they could actually see those aggregates forming.
Exactly. So they had multiple lines of evidence confirming that oxidation was indeed pushing these dynamic SOD1 droplets towards a more solid aggregated state, the kind of state we see in ALS.
This is all so fascinating. It's like we're watching this protein transform right before our eyes. But did they look into whether specific parts of the SOD1 protein are more vulnerable to this oxidation-driven transformation? Like, are there any weak points in SOD1's structure?
That's a great question. And yes, they did. They focused on two particular amino acids in SOD1, Cys111 or Cys111 and tryptophan-32 or TRP32.
Cys111 and TRP32. Got it.
Yeah.
What's so special about these two amino acids?
Well, they're known to be quite susceptible to oxidation, kind of like those spots on a metal fence that rust first.
Ah, I see. They're like SOD1's rust-prone spots.
A great way to put it.
So how did they actually investigate the roles of these amino acids in SOD1 oxidation and LLPS?
Yeah.
Did they just zap them with hydrogen peroxide and see what happened?
Not quite, but they did something very clever. They created mutant versions of SOD1.
Mutant versions, like with superpowers.
Not superpowers exactly, but they essentially replaced Cys111 and TRP32 with other amino acids that are less prone to oxidation.
Ah, so they were trying to protect those rust-prone spots.
Yeah. Exactly. And then they exposed these mutant proteins to hydrogen peroxide and used a technique called SDS-PAGE to analyze what happened.
Okay, back up for a sec. What's SDS-PAGE?
SDS-PAGE is a technique that separates proteins based on their size. It allows you to see different forms of a protein, kind of like a molecular sorting tool.
Gotcha. So what did they find when they analyzed the oxidized EGFP-tagged SOD1?
Okay, so picture this. You run the samples on this gel, and it separates the proteins by size, right?
Right.
And when they looked at the oxidized EGFP-tagged SOD1, they saw this distinct upper band on the gel.
A band. What does that mean?
A band indicates that the protein has been modified in some way. And in this case, the upper band was a clear sign that the SOD1 had been oxidized.
Okay, so they had visual proof that oxidation was happening, but what about those mutant versions of SOD1? What happened to them?
Well, when they analyzed the mutant where Cys111 had been replaced, that upper band, the one indicating oxidation, it was gone.
So by replacing Cys111, they essentially shielded SOD1 from oxidation at that specific spot.
Exactly. It confirmed that Cys111 was indeed a key site of oxidation.
That makes sense. What about the mutant where TRP32 was replaced? Did they see the same thing?
It was a bit different with the TRP32 mutant. The upper band indicating oxidation was still there, but another band, one corresponding to SOD1 dimers, it was missing.
Dimers, what are those?
Dimers are two SOD1 proteins bound together, and the fact that the dimer band was missing suggested that a different type of oxidation was happening in this mutant, possibly involving the formation of a covalent bond between SOD1 monomers through that oxidized TRP32.
So both Cys111 and TRP32 seem to play a role in SOD1 oxidation, but in different ways. That's really interesting. But how did these mutations actually affect the liquid-to-solid transition? Did they make a difference?
They absolutely did. The researchers found that mutations at both Cys111 and TRP32 altered how oxidation impacted SOD1's ability to form those dynamic droplets. It really solidified their involvement in that crucial liquid-to-solid transition.
So it's like these mutations are tweaking the controls of SOD1's phase separation behavior.
That's a great way to think about it.
making it more or less susceptible to oxidation-induced aggregation.
Mm.
But what about the mutations in SOD1 that are specifically linked to ALS? Do they also affect this liquid-to-solid transition?
That's the million-dollar question, and it's what the researchers investigated next.
It really is mind-blowing, isn't it? We think of proteins as these, static things, like they have a structure and that's it. But this research shows just how dynamic they really are and how these shifts in their physical state from liquid to solid can have these huge consequences.
It's a whole new way of thinking about proteins and their behavior, that's for sure.
And this liquid to solid transition, this delicate dance between, order and disorder, it seems to be a major player in SOD1's role in ALS.
It really does.
And, if similar transitions are happening with other proteins involved in neurodegenerative diseases, it could completely change how we approach treatment for these conditions.
Imagine, right, developing treatments that specifically target this liquid to solid transition, potentially preventing or even reversing that aggregation process in all sorts of diseases.
It's pretty incredible to think about. It feels like we're right on the edge of like a whole new era in understanding and treating these devastating diseases.
I think so too.
And it all goes back to this, fundamental exploration of how proteins actually behave. It just shows you the power of basic science, of really digging into those molecular details.
It's those tiny details that often hold the biggest secrets.
They do. So let's take a minute to recap our journey today. We started with SOD1, a protein that's usually a protector against those harmful free radicals.
Our cellular superhero.
Exactly. And then we explored this really cool process of liquid phase separation, LLPS.
Where SOD1 forms those dynamic liquid-like droplets in that super crowded cellular environment.
Like a bustling city, as we said. And then we saw how oxidative stress can actually disrupt that balance. It pushes those droplets toward that more solid, aggregated state.
That's when things start to go wrong.
Right. And we talked about the impact of specific mutations in SOD1, especially those linked to ALS.
Yeah.
They really seem to accelerate that whole transition towards aggregation.
Those mutations can really tip the scales, that's for sure.
This research has just given us such valuable insights into the molecular mechanisms behind SOD1's role in ALS. It's like we've been given a glimpse into this intricate dance of proteins happening inside our cells, and how even the tiniest changes can lead to, such devastating consequences.
It really highlights the complexity and the fragility of our biological system.
It does. And as we wrap up this deep dive, I'm left with, a sense of wonder, a real appreciation for just how complex and beautiful that molecular world is.
It's an amazing world down there.
It is. There's still so much we don't know. But with each new discovery, we get a little bit closer to understanding these intricate processes.
And closer to finding new ways to intervene and potentially treat these diseases.
Exactly. It's been a truly incredible journey, and I want to thank you for guiding us through this complex research with such, clarity and enthusiasm.
The pleasure was all mine. I always love diving into these fascinating topics.
And to our listeners, thank you so much for joining us on this deep dive into the amazing world of protein behavior and its connection to neurodegenerative diseases.
We hope you learned something new today.
We do. And who knows? Maybe this research will spark some new ideas and inspire future breakthroughs in the fight against these conditions. Until next time, keep those brains buzzing.